Hi, everyone. It's Amanda. And since this month's newsletter, we're talking about food and we're exploring a little bit the relationship between food and cancer, I wanted to spend a few minutes just talking to you about the role of food and particularly phytochemicals in specific food and how we know that they can regulate processes that can prevent the progression of cancer. So let's take a look at that. So I'll let you know that this is a very short presentation, but there's some parts of it that are quite technical. And that's the nature of when we're talking about genetics and nutrigenomics and how food interacts with our genes. It's not an easy science. So just know that there's some technical parts. I'm going to make them as simple as possible, but hopefully you'll find this fascinating as I navigate through this. And in this short presentation, I'll be actually showing you slides that I actually present to fellow clinicians when we're looking at this food gene conversation, very specifically as it pertains to cancer. So we want to know from a 100-foot view, how do phytochemicals in food work against cancer? So phytochemicals are also called bioactives. And what they are these non-nutritive compounds that we find in food that have a role. When we say non-nutritive, we mean they don't have a caloric value. They're not minerals, they're not vitamins, but they appear to play some health conducive role. They have this property. And the property they have is really as a signaling molecule that initiates processes in the cell. So think of them like a signal is the simplest way to think about them. So specific to cancer, I created this little chart from this research paper. So we know that these phytochemicals can act as blocks against initiating uh, tumor growth. So we call this tumoro or tumorigenesis. So they act as blocking agents. They can, if we go down here, they can prevent the sort of activation of cancer, but they can also work once cancer has been initiated to suppress the further promotion or progression of cancer. So they act again like a suppressing agent. If sort of the horse got out of the stable, they can kind of play catch up, if you will. They can stimulate the detoxification of toxins and carcinogens and help to remove them from the body. That is a process of detoxification, which I'm not really going to be talking about here, but some of you are familiar with. And basically, some phytochemicals can do multiple duties, and I'll actually show you that. So the question is, how do they do this? So, complicated slide you're going to see a couple of times. And what I want to show you here is a fact that's listed on the right-hand side, and that is that in cancer, we see the reversal of specific processes. In cancer, tumor suppressor genes become silenced. And here they are. These are silenced tumor suppressor genes. Think of that name. It means their job is to suppress tumors, which result in cancer. And in cancer, they're deactivated. In other words, they can't do their work. And in cancer, the reverse is also true that oncogenes are activated, shown in green. So oncogenes, so pro-cancerous genes become activated, silencing genes are deactivated, which is the reverse of what we want. And what is happening here is we have these modifiers, which you're going to learn about, which are sort of they're released, they're out of the gate, doing their dirty duty, if you will. And as you can see, our dietary phytochemicals or polyphenols can block the action of these molecules. So the question is, what are these molecules and how do they work and how do we block them? Which food? And this comes down to something called epigenetics. So epigenetics, let's read the first bullet here. Epigenetics, the definition is it refers to heritable changes that are not actually embedded or encoded in the DNA or in your genetic materials itself, but they play an important role in how gene expression is controlled. So it's not your DNA, it's modifiers to your DNA that, if you will, are above the gene. So they're not the genetic material. They're doing something that influences how that genetic material is handled. And examples of epigenetics, which we're going to look at very quickly, are DNA methylation and histone modification. So I may have already lost you, but hang in there because it's important to understand what these processes are, because that explains how food works. And so we know, as you can see in the point of me talking to you today, is that there is evidence in humans to suggest that diet and environmental processes or factors directly influence epigenetic mechanisms. In other words, what you eat and the environment that you live in can directly impact this turning on and off of these processes, these epigenetic processes that impact your health. And in this case, we're talking very specifically about cancer. And the researchers in this paper, you can see the source below, have made it very clear that we know examples of food where the molecules or the polyphenols in these foods are actually working to turn off these damaging epigenetic modifiers. Examples include green tea, turmeric, soybeans. So you may have heard of genistein, for example, broccoli, and the phytoactive or the phytochemical sulforaphane, or even isothiocyanates. These are all examples of molecules that can influence the epigenetic process. So what are we talking about then with epigenetics? So you probably heard of methylation for many, many, many different reasons. We're only going to look at methylation as it pertains to how genes are turned on and off. What does it mean? Complicated looking diagram. What you want to know is something called methyl groups have the ability to attach to your DNA and deactivate gene expression. In other words, methylation may be quite important to silencing oncogenic or cancer forming genes. This CH3 is a carbon and 3 hydrogens. That's a methyl group. And basically what you're seeing is a molecule which is transformed by adding a methyl group. And in the same way, what the diagram is showing is we can add this same methyl group to a specific part of your DNA, a CPG island, and methylation can inhibit, silence, or shut down gene expression. So one of the reasons we're very interested in the methylation cycle is to produce these methyl groups, because one of their roles is to silence genes. They're also very, very important for many enzymatic reactions in the body, but we're talking about cancer today. So methylation is an example of an epigenetic switch, if you will. It's a gene silencer. So we want that process to work. The other epigenetic switch, which is a little bit more complicated to understand, is something called histone modulation. And histone modulation or modification can cause the silencing and unsilencing of genes. So the reason I have a picture of rope here is I want you to imagine this as a coil of rope. So if any of you sail or have sailed, or if you've taken a cruise or a ferry somewhere, when the boat or the ship docks, you will actually see the ropes that tie down the ship, if you will, or the boat, and they're coiled in a perfect pile, if you will. I don't know a lot about tying, maritime knots or coiling ropes, but suffice to say that when the rope is coiled, it's not going anywhere. It's kind of an anchor. So think of your genes as being tightly wrapped by a coil. And when they're tightly wrapped by a coil, they cannot be read. They're in a silent mode, if you will. It's the same way of thinking about reading. You can't read a book if the book is closed. But if you open up the book, you can read it. If you open up your DNA, which is a process that histone deacetylase or HDAC molecules do, then the DNA can be read. So think about that. We want to keep oncogenic or tumor forming genes closed. We don't want them unraveled to be read. On the other hand, we want to unravel tumor suppressor genes, so genes that suppress cancer. We want those to be read. And this is all a process of histone modulation. So certain enzymes that are involved in histone modulation can open and close the book or coil or uncoil this rope, okay? And so we know that in this process, and it's complicated to understand, but if you get an idea of this opening and closing of books, we know that specific molecules or specific phytochemicals, such as sulforaphane, not only is it potent inducer of detoxification to help you get rid of carcinogenic processes or molecules or compounds, but sulforaphane can also dynamically inhibit histones, the unraveling of histones. So again, you want to inhibit the reading of genes that could be carcinogenic, but you want to open up genes that help suppress the cancer process. So this is an example. That's an example of a molecule that can help us do this. So histone modulation and methylations are epigenetic processes or switches, or if you will, that are involved in cancer promotion and cancer suppression. So if we go back to our little diagram that you saw earlier, remember that we want to keep tumor suppressor genes activated, but we want to deactivate genes that are carcinogenic. And it's a little bit difficult to get your head around with this slide, but as you listen to this recording, listen to this little presentation, in the end, your head will your mind will get around this. So the bottom line is dietary polyphenols or phytochemicals such as sulforaphane or dietary can actually get in there and block the processes by which carcinogenic genes are activated. And you can also block the processes or the epigenetic modification by which tumor suppressor genes are silenced. So here, for example, you see this, we don't want to silence tumor suppressor genes. We want to open them up to be read. The HDAC or the histone deacetylase can actually block the reading of these specific genes. So we want to block an HDAC inhibitor so that tumor suppressor genes can be read. The flip is we want to block the modification, the epigenetic modifications that are actually activating carcinogenic genes. So if all that doesn't make sense, just know that we can get in there with dietary polyphenols and reverse actions that are counterintuitive to our biochemistry. We can reverse actions that might otherwise lead to cancer. Now, I think it's really important to say here that if I'm working with someone who has cancer, I do not work alone. Working with cancer requires a team of oncologists, doctors, dietitians, and other experts who work with cancer. So please don't think that if this is you, like just kind of get in there and start playing around with, different foods. Each one of us is different. Our genes are different. How we may tackle your case is completely different from the next person. And this is where genetics is so exciting, because we're literally able to see into your biochemistry through your genes and look at processes that may be contributing to the imbalances that are resulting in cancer for individuals. And we may be tackling a biochemical system that's far removed from what may be a logical process for cancer. If that doesn't sound clear, what I mean there is we may be looking at detoxification. We may be looking at aberrations in how you handle folate, how you create and repair DNA. So cancer is not just a case of plugging in dietary polyphenols or phytochemicals, but at least here you can see how we're literally manipulating processes by which we're opening and closing or silencing or activating genes. So the question is then, well, which of these foods? So this is kind of a neat visual, if you will. And what you're seeing here is a collection of foods here that have a known capability to modify those histones. And then you have another set of foods that is involved in DNA methylation. Remember, DNA methylation can silence genes. And then if you put these two together, you see that there's some overlapping foods that are epigenetic modifiers. They have both the capability to modify histones and to methylate DNA. So you're recognizing probably quite a few common foods here. And the key is that, yes, of course, we want to bring in these foods if if our team agrees with them, or even if you want to stay healthy and cancer isn't even a part of your history, your family history. These foods, you now know, they contain information that can help modify how your genes are behaving. They have capabilities far beyond just this epigenetic switch or these switches that I'm talking about. I think too, one of the things that's important is eating the foods is one thing, but if we're actually dealing or working with cancer treatment, the amounts you may need and the concentration could be completely different from one person to another. For some people, maybe there's enough information in the food, but other people may need some form of more concentrated sources of these different phytochemicals. But at least you can see our basket that we're working with for this food gene connection. And so I created this little chart for the clinicians that I'm working with. And this is the chart that, based on science, we know that these particular food groups contain foods that are rich in the specific bioactives that we know have this food gene connection and this epigenetic connection. So this is really a summary of the science, if you will. And again, we're not talking about specific quantities here, more kind of knowing what to put in your basket, because the basket of ingredients contains these very specific polyphenols or phytochemicals that we know through science have demonstrated epigenetic capabilities. I think another thing that's worth mentioning here is a lot of this research has been done in lab settings. It's been done with animals, it's done with human cells, and there are now studies with humans ourselves. But suffice to say that not all of the phytochemicals that you're looking at have been demonstrated in human studies. However, you can also look at these foods and say, well, they're everyday foods. If there's nothing special, there may be some little notes I might make about the specific sources of the food, how you prepare the foods, other applications for the foods. But you could fill your basket or your grocery cart with any of these foods and know that you're providing information for your genes. So hopefully this has given you a little bit of an overview of how food works we call it in a nutrigenomic capacity, to help turn on and off processes that can be carcinogenic or can prevent the promotion and growth of cancer. So I hope that helps. And if you have any questions, feel free to send me an email, amanda@genomickitchen.com. It may take me a couple of days to get back to you, but I'm more than happy to give you some insights or answer some of your questions. Thanks so much, everyone.